Hello everybody, so we are going to get a head start on bio this semester, starting with our slideshow on evolution. I hope you recognize the person in this photo and the birds that are on the right hand side, and if you don't, this is Mr. Darwin and his theory of evolution regarding the Galapagos finches. Okay, so quick vocab check. We're going to look at the different levels, so starting with species. A species is all members of a similar population that can naturally interbreed and produce viable offspring. Keep that in mind, viable offspring. A population, which is the next step up, is all the members of a single species within a particular environment. A community, which is the next step, is all of the different populations within a particular area that interact with each other. An environment is the area and conditions in which an organism lives, and then an ecosystem is all of the living, biotic, and non-living, abiotic, things within a particular area. The difference between an environment and an ecosystem is that an environment is a lot broader than an ecosystem. An ecosystem is more specific. They're often used interchangeably, but keep in mind, environment is broader because it's a larger word, and ecosystem is more specific because it's a smaller word. That's not the exact reasoning, but that's a way to help you remember it. So what is evolution? Evolution is the change in a species over time, which means phenotypes. This is the old definition, which Mr. Mason does not want us to remember, but it's the short, quick way to think of it. There are different types of evolution. There's divergent evolution, which is the process by which groups of related organisms evolve in differing directions from a common ancestor. So if you look to the right-hand side, the tiger, and then I'm assuming that's a bobcat, they have a common ancestor, but they've completely evolved separately. They don't look anything alike. Convergent evolution, on the other hand, is the process by which different groups of organisms evolve into similar forms, which means they don't have a common ancestor at all, but somehow they evolved to have similar features. So if you look onto the right-hand side again, both the bat and the dragonfly, they don't have a common ancestor, but they both have wings. They both fly. And then our modern definition of evolution is the change in allele frequency in a species over time. So this is genotype. So instead of looking at the phenotypes of an evolution, we're now looking at what we can't necessarily see with our eyes. We're looking at genotypes, alleles, how our genes and alleles change over time. Moving on. So we talked about evolution, but let's get a little bit deeper into it. So there's macro and microevolution. Macroevolution is when we have major changes to a species, but it takes a long time. But it results in a new formation of a new species, and this is called speciation. We also have microevolution, and this is small changes in a species, but it's in a really short time period. And then we're also getting some sort of change as well. And microevolution is primarily based on allele frequency. So if we look to the right-hand side, we'll see some examples of those as well. So again, macro, big, long time, micro, small, short time. So what is the evidence that we have to prove that evolution is a thing? Well, our biggest thing is fossil records, the complete collection of all fossils found on Earth. That is our fossil record. Fossils are the petrified remains or impressions of previously existing organisms. So they show our potential changes over time in a particular species, and they also contain extinct species. So those are the species that no longer exist, but we have evidence to show that they did exist at one point. And because we have evidence of these extinct species, they can show how other possible organisms branched off from a common ancestor, which again supports our theory of evolution. And then some ways of telling, well, how old is this fossil? How new, or which ancestor do we think it could be within the same time period from? We have two different ways. We have relative dating, which is based on strata. So relative dating in plain terms is what layer of dirt did you find it in? The closer to the surface, the closer the strata, which is the type of dirt, the closer it is, the newer it is. And then if you find it really deep within the earth, that's going to be older. So we know that it happened a long time ago. And then we also have radiometric dating, and this is utilizing specific isotopes to kind of identify that rate of decay. And the most common isotope we look at is 14 carbon. And the reason we look at carbon is because, if you remember, all living things are carbon-based. Life is carbon-based. So that's why we use 14C. Radiometric dating can also be referred to as carbon dating, again, because life is carbon-based. So if we're looking at life, we know that they're going to have some form of carbon left over. So here are some photos of evolution. So if we look on the right-hand side, this is a horse. And at the very top is what we currently have today is Equus, the horse. And then if you look all the way back, we have Mesohippus, which is our latest equine variation. On the right-hand side, we have chimpanzees, gorillas, and then Homo sapiens, which are humans. And there is a theory that we evolved from them. Who knows? I'll let you decide for yourself. Now, other evidence of evolution, we have different structures, and looking at these structures, we can see, are you common, are you not? What do you have in common? So our first thing is a vestigial structure. So a vestigial structure is an inherited structure that has lost most or all of its original function. So surprisingly, snakes actually have legs. Now, you might think snakes don't have legs, Ella. Well, they actually do. They have vestigial structures, and these are bones, and they're little legs, but they've been reduced over time because they've lost their functions. Snakes don't use them anymore, but that doesn't mean they're not there, or it doesn't mean that they weren't there at some point. Next, we have homologous structures. So these are body parts with similar structures or features, which suggest that there was a common ancestor. And they may or may not have similar functions. So an example to this of the right, we have the human arm, a cat leg, a whale fin, and a bat wing. All of these, if we look at the bones, they all have the same bones. They might not look the same, but they are the same bones, and they have similar functions, which suggests that there could have been a common ancestor between them. Humans use their arms for movement and hands to grab things. Cats, again, use their legs to move around and grab things. Whales move their fins to move through the water, and bats use their wings to fly and grab things as well. And then we have the opposite, analogous structures. So these are structures that are not related. They do not have a common ancestor. So these are structures in non-related species that have similar functions, but they evolved independently. And this is typically a result of similar environmental pressures. So again, looking to the right-hand side, we have a bat, a bird, and an insect. They all have wings, but they're not related. However, their conditions require them to fly. So they have that environmental pressure of the need of flight. So they have wings. And then if you look to the bottom, we have a shark, a penguin, and a dolphin. We have a mammal with the dolphins, we have a bird with the penguins, and then we have a fish with the shark. They all have flippers or fins, as we could call them, but they're not related. They're completely different species, but they all have that similar environmental pressure of the water. Penguins go in the water, in case you didn't know that. They do. They swim in the water, they catch their fish. So they have that environmental pressure, which is why they have those flippers. Next, we have embryonic development. If you look to the picture on the right at the first line, it's kind of difficult to tell what species is which. What am I looking at? But as you continue down and the embryo develops, then we can start to see some differences. So embryonic development is where multiple species have similar processes and structures during the development of their embryos, which suggests a common ancestor. So take a look at the photo to the right and try and tell me which one is which at the start, and then tell me which one is which at the end. You can clearly tell, and we can see the changes throughout the process. We can also see the similarities. All right, next we have genetics. Genetics is a huge thing within our modern technology. So modern genetics show the relatedness of different species through the presence or absence of a particular sequence. So if you look to the right-hand side, we have the warbler finch, the woodpecker finch, and the ground finch. And then if you look at the sequences, they have a lot in common, which suggests and proves that they came from the common finch. Now, speciation. Speciation is the formation of new and distinct species resulting in the splitting of a single lineage into two or more genetically unique lineages. So there's a few different ways that this can happen. So we have allopatric speciation. So this is the result from a physical separation of populations. So in the example with our fish over here on the right, all three of these lakes, Lake Victoria, Lake Tanganyika, Lake Malawi, they all used to be one whole lake. All three were together, but over time there was erosion and they became three separate lakes. They are physically separated. They're fish. They need some sort of waterway to get interconnected, but they don't have that anymore. So they're going to evolve separately. That's an example of allopatric speciation. So dispersal, which goes under that type of speciation, is where members of a species move to a new geographic region. So this could be with the Galapagos finches. They moved. And then we also have vicariance, which is where we have environmental factors that cause separation of the same group of the same species into separate groups. So in this example, we typically see this with some sort of natural disaster. So there could be a volcanic eruption that causes a lava spill and totally separates a forest with lava, leaving one population of deer on one side and the other population on the other, and now they have to evolve separately. So that's going to cause some differences. And then we also have sympatric speciation, and this occurs in the same geographic location. So all the previous examples were different locations, but this one, same geographic location. So we have gradual speciation. So this is slow and steady changes that lead to eventual changes in a species from the accumulation of small genetic changes. And this can happen within large populations most commonly. And then we also have punctuated equilibrium. And this is where we have a sudden burst of an evolutionary change, but it's separated by a long period of time of no change. So you might see a bird looks one way, and over the next few generations, it looks pretty much the same. And then all of a sudden, the next year when they reproduce, they look totally different. That's an example of punctuated equilibrium. It happens like that, but it's been changing under the surface over time. Gradual speciation, we're going to see small changes, and you're not going to notice them as much when they're happening, but if you compare the beginning to the end, you can completely see the difference. Adaptive radiation. So this is speciation that results from the evolution of a single common ancestor into several new species, and this is heavily based and influenced by an ecological niche. So again, with the examples of the lakes. Each of those lakes had their own specific niche, and so one lake was most likely deeper, one could have been more shallow, so some of those fish were more adapted to darker waters because the light didn't penetrate as deep, whereas the more shallow lake, the fish weren't adapted to darker waters because they didn't have that depth and the sunlight was reaching them. So that's going to cause differences. Next, we have the development of the theory of evolution. So where did this come from? How and where does life fit into this, and where do life forms come from? So we have key observations of life. 1, there exists a high level of diversity among all forms of life. There's so much diversity. 2, there are shared characteristics in all living things. We all have something in common. Carbon-based. That's one example. The need of energy sources, food, water, shelter, possibly warmth. All of those are shared characteristics. That doesn't mean we have to look at the same or have the same genetics, but we have some sort of characteristics in common. And then 3rd, organisms are well suited for life within their unique environments. If I took a polar bear and I put him in the desert, he's not going to survive because that's not his unique environment. His unique environment is the cold Arctic. That's an example of a unique climate. Charles Darwin, we talked about him on the first slide. He is an English naturalist, a geologist, and a biologist. That's a lot. This guy loved nature. He is most known for the Galapagos Islands and what he observed there, and what he observed was variations among individuals within the population, specifically Darwin's finches. He also released a book called On the Origin of Species, and this goes over like modifications, the foundation of modern theory of evolution, and he also proposed that species evolve over time through the mechanisms of natural selection. All right, Charles Darwin's primary observations. 1, individuals within a population usually vary in their inherited traits. This is called variation. 2, every species tends to produce more offspring than can be supported by their environment, which most don't survive or reproduce. But the reason this happens is because they already know that some of the kids are going to die from disease, be eaten by predators, or not survive a harsh winter. So if you produce more, then you have a higher likelihood of some of them surviving, reproducing, and carrying on that species. Next, he also had inferences. So those who inherit traits that give them traits allow them to survive, reproduce, and then leave more offspring. But this creates an inequality which leads to higher levels of favorable traits within a population over time, which means natural selection. Some are going to have favorable traits, some are not going to have favorable traits. But the ones that are favored are going to reproduce and have more. Therefore, the ones who aren't favored are going to decrease in their numbers. And this leads us to natural selection. Natural selection is the primary mechanism that influences evolution. Essentially, it's survival of the fittest. Now, when we think fitness, nowadays we think strength, muscles, working out, Luke in the gym. But that's not what fitness is in this sense. Fitness is the ability to survive and reproduce in a particular environment. If you took a polar bear out of his environment, he's not going to be able to survive and reproduce in the desert. That's why it's in a particular environment. So again, survival of the fittest is where individuals with the highest level of fitness will survive, reproduce more offspring, and pass on their traits. But this also leads to adaptation, and adaptation is where specific heritable traits increase fitness. And then this applies to the organism as a whole, not just a specific trait. Darwin's finches are one of the best examples of natural selection. So he observed that there were similar species of finches, 18 to be specific, but they had distinct characteristics on the Galapagos Islands. And these variations were observable between islands and overall environmental conditions. So Darwin proposed that they may have all come from a singular species that originated from the mainland and then migrated to the islands. And then each different environment, aka the different islands, had different traits that they favored. So over time, those traits became the dominant ones, leading to the development of the separate species. So this leads into the descent with modification, result of natural selection, and adaptive radiation. So selective pressures. The selective pressures are environmental conditions that favor particular variations over another. And we have 3 different types. We have stabilizing selection, which is going to be our first photo on the right, which is where environments favor the average phenotype and select against the extreme variations. Extremes are left and right, and our average is going to be in the middle. So stabilizing is going to pick the middle. Directional selection is where our environment favors phenotypes at one end of the spectrum or the other, and selects against phenotypes that are average and at the other end of the spectrum. So they either favor right or left, not the middle. And if it's right, it doesn't favor middle or left, and it's the left, it doesn't favor middle or right. And then diversifying selection is where our environment favors phenotypes at both ends of the spectrum and selects against the average. So they like both right and left, and they don't like the middle. Mr. Mason said specifically, he wants us to note these photos and in comparison to their definition. So if I said stabilizing selection and I showed you the 3 photos, you should be able to point out which graph goes there, and vice versa. If I gave you the graph, you should be able to tell me which type of select pressure it was. Genetics, allele frequencies. This is the rate at which a particular allele appears within a population. And then we have our gene pool, which is the totality of all the genes available within a population. This leads us to the founder effect, which is a significant allele frequency change in an isolated portion of a population. And again, this typically results from some form of like natural disaster, bottleneck effect, migration, immigration, so moving to and from different places. So next is our Hardy-Weinberg equilibrium. So this is a completely theoretical frequency stability idea, which assumes that pure chance, large populations, no mutation, no external influences, no natural population, no nothing, entirely chance. So let's be realistic. No natural population is going to meet these requirements, but that's the whole point of this, because if we have something that's only by chance, then we can compare it to what's actually happening in nature, and then it's useful for evaluating the allele frequencies within a real population because it eliminates chance as the source. Now moving back to genetics, genetic drift. This is where our allele frequencies change that result from chance. So again, chance, genetic drift. Alleles change from chance. Our gene flow is where our alleles move in and out of a population because of the migration of the individuals or the gametes. Bottleneck effect is a natural event that eliminates a significant portion of the genome, and the surviving alleles serve as the population's genetic structure moving on. Mutations. Mutations are the random changes to the gene sequence. So it may or may not impact the phenotype. Most mutations are harmful, but some could be positive. It's very rare that we see that though. And some might also have no effect or a small effect, but most of the time we see them as harmful. But they're not passed on because they are so harmful. And the mutations that increase fitness can become heritable. But there are also some negative mutations that also are heritable. An example of this would be sickle cell disease. Sickle cell is not a good thing, but if you have sickle cell disease, then you're immune from getting malaria. So there's some benefit to it. Moving on to biological isolation. So we have reproductive isolation, which is when 2 species Moving on to biological isolation. This is where we have our reproductive isolation. So when 2 species are prevented from interbreeding that could otherwise do so, biological isolations can lead to specialization. So one form of this is pre-zygotic isolation. So this is reproductive isolation that prevents the formation of a zygote through fertilization. So examples of pre-zygotic isolation is going to be our ecological isolation. So this is our habitat isolation, which means interbreeding could occur, but the 2 species live in different areas or occupy different niches. So think tigers and lions. Lions occupy the savanna in Africa, and tigers occupy Asia. They're on 2 different continents. While they could interbreed, they're not near each other, so they're not going to. And then we also have temporal isolation, which is where the 2 species could reproduce but the 2 species are reproducing at different times. So they could reproduce at day, it could be seasonal, it could be once per year, twice per year, so many different possibilities. But you could reproduce, but since they don't align, they don't reproduce. You have behavioral isolation, which is where the different behaviors prevent the mating. This is typically from different mating rituals. And the example from the amoeba sisters, one bird is singing country and the other bird doesn't like country. They're not going to mate. We also have geographic isolation, which we saw this with our finches and the Galapagos. It's where our physical barriers prevent mating. So rivers, mountains, oceans, or you live on one island. You could fly to the other island, but if an animal was solely isolated on the island and they couldn't swim, they're not going to be mating with another organism from, say, the neighboring island because they can't get to it. Our mechanical isolation is where there's structural differences that physically prevent mating or fertilization. So think flowers and our pollinators. If there's a pollinator that doesn't have the structural beak or ability to get the pollen out of the flower, it's not going to help that flower reproduce because it physically doesn't have the structure to do that. Chemical isolation is where there's chemical differences that prevent the fertilization. So if there's something that's too acidic, well, you could reproduce. The acidity itself is going to kill off any possibility of that fertilization. So the reproduction is not going to continue. Now we have our post-zygotic isolation. So this is after our zygote has been formed and the reproductive barriers occur following the fertilization. So one example would be a hybrid. So this is where the offspring results from the interbreeding of two different species, but it doesn't produce viable offspring. So this is what we talked about before when a species can produce viable offspring. If the hybrid is sterile and can't reproduce, that's not viable. So on the right hand side, the example that we have is a mule, a donkey, and a horse, but they can't reproduce. And they never will, sadly, because they're kind of cute. Anyway, hybrid inviability. This is where hybrids are produced, but they're not able to survive to maturity to be able to reproduce on their own because there is some issue and they physically do not reach maturity. 2, hybrid sterility. I mentioned this one before. Hybrids are produced, but they cannot reproduce themselves because they're sterile. Hybrid breakdown. The first generation of hybrids, F1, are able to reproduce, but the next generation of F2 cannot reproduce. And we typically see that in a lot smaller organisms like unicellular organisms. We don't normally see that in our bigger organisms like the mule or humans or any other type of larger multicellular organism. Other factors that influence it are competition. So limited resources that lead to competition among and between populations can result in significant changes. If there's only one food source available, but there's multiple species fighting for that food source, the one that is more fit to secure that food source is going to outcompete the others. Sexual reproduction is another factor, and this is where it results in unique individuals that can cause random shifts in allele frequencies. So the sexual reproduction is creating something new, something different, but because it's new and different, it's creating these allele and genetic shifts that's going to affect things later on. 2 is mating choice. Some species choose their mates based on a variety of characteristics, and this can greatly influence the flow of genetics within a population. One key example of this would be humans. There are some humans that only want to date or marry the hottest person in the room. And so if they're only dating the hottest person in the room and say that hot person's really dumb, then their reproduction is not going to be intelligent because there's nothing there. They're only hot. It's going to be a hot baby, but it's going to be a dumb baby. But they chose that mate. So that's on them. Where on the other side, if 2 intelligent people reproduce, then their child could be very intelligent. It might be ugly, but it could be intelligent. Or they could be hot and intelligent, and then you have a hot and intelligent baby. So there's so many different possibilities, but it's the fact of choice that is influencing the variation. Now, we have some issues and controversies. The first one are gaps in our fossil record. We have some significant gaps that exist between our proposed related species, and so this is going to cause some issues. It's especially true for the formation of new species with no previous ancestral forms that have been found. So people argue, how do we know that this is a new species if we have no evidence to support any previous ancestors? 2, the missing link. It hasn't been found conclusively to prove that human evolution came from one species or another, and then there's other species as well that have this. Because there are missing links, we don't know exactly how they connect. We can see similarities, but we don't have that full picture to finalize our image. And then 3, this may be the result of fossil rarity. There are some fossils that are just so rare that we don't have them, and that's going to cause issues with our record because it causes them to be incomplete. But the rarity can also be influenced by the fossilization process, environmental factors. There could have been certain fossils that lived in one environment, but there was a volcanic eruption and it completely destroyed any fossil remains that were there. And so now we have no record of them ever, and we're not going to. So there's so many different factors that can lead to this, but it causes those issues and controversies because people can't fill in that gap. And if people don't have the evidence to do that, I'm going to cause some issues. 2, the Cambrian explosion. So this was a geological event where nearly all major animal body forms rapidly appeared in the fossil record. So this was from 551 to 510 MYO during the Cambrian period. So a long time ago. But because of this, there's no transitional species that's present. We have all these fossils, but nothing that's present. And then this could be the result of an ancestral species having like a soft body and not being able to be fossilized. But again, we have some evidence, but we're lacking some. Another thing about the explosion is that it includes a sudden appearance of highly complex structures, such as like compound dyes. But again, since we don't have those connection points, it's hard to correctly identify where and when it came from. Next, life only comes from life. That is literally one of the foundational units that we learned last semester. The foundation of life from the cell theory and the characteristics of life. All living things do not spontaneously appear. The initial source of all life and its forms come from life. And so if its form is not scientifically found or proven, we're going to have some issues and controversies over that. And then again, it's a matter of time, because as time progresses, we're going to lose more and more things. Sure, we're going to have more technologies to try and help us find those, but we're still losing a lot because we don't have the resources right now, and things naturally are going to be lost to time. And then next we have the evolution versus creation. It's a huge rift between religious faith and evolutionary theories. There are a number of religions that have creation stories as a part of their faith, like Christianity. We have the story of Adam and Eve being the first two humans and continuing on from there. Nowhere does it say that we evolved from apes, yet there are some theories that say, based on evolution, we evolved from apes. Well, that's not what Christianity tells us. There are a number of other theories as well from other different religions. But again, it's going to cause a lot of differences because people are going to see different views in science and faith, and which one do they believe in more. And there's evidence to support both theories, but again, it comes down to the individual. I hope this video was helpful to all of you guys and it helped you study. If you have any questions, please let me know, and I will continue to update you with more videos.